The talk I've been invited to give seemed to me a little bit confusing, and I talked with Richard and I prepared something based on what we do, our history, how we're funded, where we're going, that type of thing. I hope it's satisfactory. So I just thought I'd introduce the people first in the group. Here we are. This is me. This is Deepika Devaner. She's working on the protein biomarker study, the development of the diagnostic test. Beverly, Robert and Joanne are all working on the gene expression studies. John is our research nurse who helps with sampling patients and input of clinical details. And David is based at Bristol with the psychopharmacology unit there, and he's involved in recruiting CFS patients and patients with depression. The use of patients with depression is important because we want to be sure that our gene expression signature is specific to chronic fatigue syndrome and doesn't occur with other diseases. We're also recruiting patients with osteoarthritis, rheumatoid arthritis, several other infectious and rheumatic diseases for that purpose. We do clinical work, and at the moment, this is just me. I work in infection and medical microbiology at St. George's Hospital. Soon we plan to start a CFS clinic at St. George's Hospital in the Department of Rheumatology. We do research, obviously. We teach, and various members of the group give lectures on their work locally, nationally, internationally. We teach within the hospital on infection and medical microbiology, infection control, etc. And there's management, so we manage people, we manage budgets, etc. So in the research, we aim to develop an understanding of the pathogenesis of chronic fatigue syndrome, and the way we're doing that is by studying gene expression. We use microarrays and real-time PCR to study individual genes. We want to develop a diagnostic test for chronic fatigue syndrome, and the way we're doing that is using mass spectrometry to analyze the proteins in serum samples. Particularly, we're using cell DPC for that. We want to develop treatments for chronic fatigue syndrome, and we plan initially to base that on our gene signature, which we now have, and there are several experimental treatments suggested by that signature. And finally, I'm involved in another study on the rapid diagnosis of infection in infected patients at St. George's Hospital. So the gene expression studies then are principally involve analysis of human gene expression, but we have also a study which is focusing on the role of virus gene expression in chronic fatigue syndrome, and certain features of our human gene signature suggest that a virus might be involved in the pathogenesis. And we have another study based on screening of total RNA samples, which has picked up several unique signatures of human and other viruses. I've already mentioned this. We're using mass spectrometry to identify proteins in the serum of people that are either raised or lowered compared with normal people and other diseases, so that we can use these to develop a diagnostic test based on commonly found formats used in clinical laboratories, such as an ELISA format or a dipstick format. At the moment, we have several biomarkers that we're developing for a diagnostic test. And finally, we're looking at cell signatures of past persistent virus infections and non-virus infections. And this is basically our attempt to get to an understanding of a possible prodrome of disease, whatever that final disease may be. It's our hypothesis that viruses that after the acute phase that persist in the body may perturb the immune system slightly, and this may be a small abnormality that persists over a very long time, and that may have an impact in development of future disease. And we have evidence for that. So day-to-day activities, we discuss on the phone with various groups in the UK and the USA and Belgium and other countries how we're going to recruit patients to the studies. These are CFS patients, normal people, and patients with other diseases that we need to confirm specificity of our signature. We receive samples in the lab, hopefully in weekdays, so we can process them on the same day. We process these. We receive a PAX gene tube for RNA isolation, an EDTA blood tube so we can obtain genomic DNA, and a clotted blood tube for obtaining serum samples. And these give us all the samples that we need at present for our studies. We then convert total RNA to complementary DNA using reverse transcription, and this is the sample that we test for gene expression. All these are then stored at minus 80 degrees centigrade, which keeps them, we believe, to be the same for at least several months. And we aliquot them so that if we need to test them, we don't have to thaw the entire sample. We just need to take one of several aliquots, and then the others remain unthawed at minus 80 for several months. We receive the clinical details of patients' controls, and these are then entered into the computer by various of our staff. These will then facilitate subsequent analysis and characterization of the patients. We test the genes using particular methods based on polymerase chain reaction, and we test proteins as well, like I've said. So we use microarrays principally to screen the samples for RNA expression. The scanning of a microarray produces this rather nice rectangle with a lot of little coloured dots, some very dim and others very bright, and this gives us the relative expression of the different genes. And we use polymerase chain reaction, or TACMAN PCR as shown here, to confirm associations with particular genes. And this shows the TACMAN plot. We've got number of cycles from 0 to 40, and we've got detection times for the test gene around 26 and 32 or something for the control gene. And it's this comparison of the test gene and control gene for one patient, for one test gene, that gives us the information we need for each subject. So in cell DPC, that's mass spectrometry, we spot serum samples or fractionated serum samples onto these protein chips. These are then put into the machine here, and they're excited with a laser, and the ions are then allowed to travel down a column, and the time it takes them to do that can give us vital information about those molecules according to this mass spectrum. And then we compare these spectra between cases and controls to detect the differences. So in this example, we have the black trace is the normal people, and the red trace is chronic fatigue syndrome people. Just to illustrate that you can see differences where these arrows are. And this is the principle of the method. So I also supervise PhD projects, one on gene expression in chronic fatigue syndrome, and another one on the cell signatures of past persistent virus infections. And these are roughly halfway through now. So more mundane tasks, we order items for the research, such as kits, such as pipettes, such as pipette tips, bottles and tubes and racks for storing samples, etc., etc. Managing people is very important, and we do a yearly performance development review to make sure people are happy, that they have no problems, that they haven't identified any problems with me, etc., etc. Managing budgets is very important. We need to make sure we're using up the money at a predicted rate. Health and safety, very important too. We're working with samples which could be dangerous. We don't screen the samples for HIV. We assume that they could possibly be infected with those dangerous viruses, and we take universal precautions as a matter of course. So more interesting tasks. We do a lot of reading, thinking, testing samples, data interpretation, discussion of results and planning experiments. And a lot of the work is done on computers. So we use computers to send emails to each other, to other groups, to coordinate sample receipt, to exchange research information, to obtain electronic research papers, to analyze gene information and gene expression data, and to submit papers to journals for publication. So like I've said, collaboration is a critical aspect of our work, and we have collaborations in Europe, in Australia, and in the USA. And I was told by my mentor, David Tyrrell, always have several strings to your bow, because he said, you don't want one important thing to dry up and then you're stuck. So we have several projects running, and I think that's very important. So more difficult tasks are getting funding, and this is based on having an idea, which is a good idea based on what is known at present. We then select an appropriate charity or donor and apply, outlining the people, the place, background, plan of investigation, duration, costs, and key signatures. And then we either succeed or fail. We are presently funded by the CFS Research Foundation, which is based in Hertfordshire, UK. And we started with £1.1 million to do this work. We're also funded by the ME Research UK. They gave us a small grant to look at the gene expression signature in Gulf War syndrome patients. We've applied several times to the MRC. On each occasion, we were invited to submit those applications, and on each occasion, we got scores typically of 9, 8, and 3. And the score of 3 was obviously from a psychiatrist who was complaining about our way of enrolling the patients, the criteria we had, etc., etc. And when we got the rejection email, on the same day we got an email, please can you call me, from the board manager. And when you called him, he said, I hope you weren't too disappointed with that, but we're still keen to fund biomedical research in chronic fatigue syndrome. So, and David Tyrrell told me something else as well. He said the MRC will never fund biomedical research in chronic fatigue syndrome because they're in the thrall of the psychiatrists. So far he's been right. So then we have to present our work at meetings, local, national and international. And that's very exciting, to meet other researchers and meet patients and discuss work and hear about other people's work. We publish papers, and this can be quite difficult in the area of chronic fatigue syndrome, similar to getting the funding. And future research, well, what are we going to do next? Well, we're going to confirm the specificity of the gene signature. As I've said, we have 80 genes in that signature, and we need to make sure that the genes we end up with at the end are ones that we can call specific. We need to look at the variation of these genes with time, so we're going to look at a subset of patients and look at them every month for a year and compare the gene levels with the presence of individual symptoms at those times. We need to look at peripheral blood mononuclear cell subsets, and this is important because so far we've only taken all the white blood cells in the blood and looked at the gene expression in those as a group, and that's a standard approach. But what we need to get a step further in understanding of the pathogenesis is to find out which of our genes occur in the T lymphocytes, the B lymphocytes, and the monocytes, and the natural killer cells, for example. We will continue with our studies looking for viruses in chronic fatigue syndrome. And this is quite challenging because there have been many studies in this area so far. So why have they not found something? Well, we're thinking a lot about that. Maybe we've got a recognized virus which is behaving in a new way. Or maybe we've got other viruses which haven't been recognized yet. And finally, we want to do clinical trials using treatments based on our gene signature. And we haven't actually done any yet. We applied for funding and we're not successful. But we're still negotiating on that, and I can tell you more about that tomorrow. So just to acknowledge the help of clinical collaborators across the world, people for study design and laboratory work, and principally the CFS Research Foundation for funding. Thank you very much.
I wasn't very clear how your volunteers were recruited and how much diagnosis they'd had before the samples were taken.
There's the chronic fatigue syndrome patients and others. Well, they're diagnosed according to Centers for Disease Control clinical criteria for chronic fatigue syndrome, which depends on exclusion of other physical and psychiatric causes of fatigue. Those were the chronic fatigue syndrome people. The normal people are screened routinely by the National Blood Service, and we ask some additional questions according to drugs they take, and if they're smokers or not. We exclude several drugs and smoking.
Yeah, thank you. Got a couple of questions which are quite general, really. First of all, the output of your research, where do you aim it to go eventually, and who's likely to use it? And secondly, more sort of curious on the genetic aspect, not knowing anything about this. I thought genes were something we inherently had as an individual. That's sort of by layman's thought. So are you testing CFS patients to see whether they have specific sort of genetic markers or whatever in comparison with normal population to establish whether or not a particular person would have a predisposition to get the disease? Is that the idea of the research? Thank you.
Is your first question, does it relate to where we plan to publish the research, or?
Well, who's likely to use it? Where you're aiming it to go?
Well, I think at present...
Is it drug companies who are likely to look at the research to develop drugs, or
yes,
doctors and other people in the field who will use it to diagnose? Or I'm just curious to know where that research like this, what happens to it? Does it just stay in a vault somewhere?
Basically, it gets published in a journal, and it's there for people to read from that point on. It gets presented at conferences like this one, and it gets discussed. And yes, it can be looked at by drug companies who can then use that to develop drugs, and we may look at that to develop drugs. It's a standard approach. The things we're doing are very standard. They're not special in any way. And your other question was, are we looking at inherited determinants of likelihood of development of chronic fatigue syndrome? We're not yet, but we are collecting the genomic DNA samples of patients and controls. There are various ways to do that, and the way we're going to do it is we're going to take those genes that we find in our gene signature and then look at the variation of those genes.
Roger. Just a quick question about your study group. If there's a high prevalence of Lyme's disease in your group?
We're not testing for that.
Okay.
But if there were symptoms suggestive of that, we would, but we haven't tested anybody for that so far.
Is it at all possible to even estimate how far away we are from some sort of drug breakthrough on treatment? Are we talking, 2 years, 5 years, or decades?
I think it depends on funding. I mean, we would like to do a study of a TNF-alpha inhibitor. We haven't yet been able to get a drug company to give us the drug, but we're now thinking about simply buying that and working that into a grant application. So, I mean, this drug has been used before, and it's been presented 5 years ago as being very beneficial in chronic fatigue syndrome. So it's difficult to answer your question, but it could be fairly quick.